All right, back for another video. What I'd like to do this time is talk a little bit about neurotoxins. And this is definitely supplementary to your understanding of how the component parts of a neuron, and especially the proteins involved with the membrane of the component parts of the neuron, will affect neuronal function. And so I think neurotoxins are just a really good way to sort of make sure that you understand the importance of voltage-gated calcium channels or voltage-gated sodium channels, or you name it. So we'll do a sort of a subset of neurotoxins, sort of assessing what their targets are and how they would affect neuronal function. Now, the key to remembering here, well, let's start back up just a second. Neurotoxins come from a variety of sources. A lot of animals and plants use neurotoxins as a defense mechanism or a way to kill their prey, or at least paralyze their prey while they kill their prey. And so they're found all across the animal and plant kingdoms. I'm going to do a subset, like I said before. I'll make a list down here at the bottom of them. Let's start with one called Tetrodotoxin. This is a really well known. It's usually abbreviated TTX. And Tetrodotoxin blocks voltage-gated sodium channels. And so, again, this is a good way to think about application-style questions, right? Where I throw some new toxin at you and you have to sort of assess the effects of it. And so, tetrodotoxin blocks voltage-gated sodium channels. Well, so you have to sort of think, well, where do we find voltage-gated sodium channels and what is their role in neuronal function? So for voltage-gated sodium channels, you need to immediately be thinking axon. Right? Axon, and to a lesser degree, the synaptic bulb. And so, okay, what's the function of voltage-gated sodium channels in the axon function? Well, the axon functions in the transmission segment or function of a neuron. In other words, we get action potentials traveling rapidly down the length of this axon to deliver a signal to our postsynaptic cells, in this case, a skeletal muscle. And so we can get all kinds of input here. I can show you other cells synapsing here and generating input. Right? We could get EPSP input here and reach, let's add this, at our axon initial segment. Right? Because that's our critical sort of beginning of where we find the voltage-gated channels. But if we block the voltage-gated sodium channels with tetrodotoxin, guess what? You could reach the threshold of those channels here and they don't open. In which case, input here, even massive stimulatory input here, would lead to the generation of no action potentials. Well, if this target cell is a skeletal muscle fiber, or skeletal muscle in general, right, then no action potentials here means no depolarization of the bulb here, which means no neurotransmitter release here, which means no activation of that skeletal muscle. And you can imagine this is very dangerous because if this causes paralysis, right? In this case, it would be what's called flaccid paralysis, because we wouldn't be able to stimulate that muscle. It can be really dangerous because if you think about it, these could be respiratory muscles, which are a type of skeletal muscle, and it paralyzes your respiratory muscles and you asphyxiate. So stay away from tetrodotoxin. Found in a lot of, not a lot, but some marine animals, including pufferfish. I think there are a few other animals, marine animals, that also produce tetrodotoxin. Okay, let's do another one. Let's do Conotoxin. So Conotoxin is produced by the cone snail. And Conotoxin blocks voltage-gated calcium channels. So in this scenario, and again, we'll focus on this neuron as opposed to this one, but you could get input to, let's say you got input to this neuron, You could get action potentials, but that action potential would reach the synaptic membrane down here and no longer cause an influx of free calcium. And remember, calcium was required for the synaptic vesicles to fuse, right? We can add some synaptic vesicles here. Right? And if this is skeletal muscle, they would contain acetylcholine. There's a lot of neurotransmitters, but at skeletal muscle, it's acetylcholine. And if we block the voltage-gated calcium channels, we don't get any neurotransmitter release. If we don't get any neurotransmitter release, again, we don't get any stimulation of this downstream muscle. And you would again end up with flaccid paralysis, where the muscle is not activated. Different mechanism, same end result. So that's the cone snail Conotoxin neurotoxin. Okay, let's do another one. Alpha-latrotoxin. So alpha-latrotoxin is produced by the black widow spider. And this is a toxin that actually formed pores in the membranes of neurons, and these pores are permeable to sodium and calcium. So if you think about it, now we could focus on this cell or this cell, but if we think about it, we open up sodium and calcium channels on this cell. The input to this cell just went up, right? Sodium and calcium would generate EPSPs, right? They tend to move into the cell down their electrochemical gradients, very positive equilibrium potentials. And we would get a larger EPSP. And again, even if it's randomly, we would either get more neurotransmitter release, or if it formed pores in this cell that allowed calcium influx, we would get an EPSP even outside of a synapse. And so we would get more action potential and sort of excessive stimulation of this neuron, more action potentials, more neurotransmitter release. And so now we have overstimulation of the target cell there. So now you end up with what's called rigid paralysis. This is paralysis where the muscle overcontracts and can't stop contracting, essentially. So you end up paralyzed, but not because the muscles are inactivated, but because they're overactivated. Okay, so that's the black widow spider venom. Let's do tubocurarine. That's usually just called curare. It's a plant toxin used on the by some Aboriginal people on their arrows to hunt animals because it's a paralytic. And what it does is it blocks acetylcholine binding. And so what it's going to do is it's going to block receptors down here on the target tissue. Right? Again, we said acetylcholine is released if this is skeletal muscle. And so the neurotransmitter, this can be activated. Neurotransmitter can be released. The acetylcholine is diffusing to our postsynaptic cell here, the skeletal muscle cell, but it can't bind. And if it can't bind, then you don't get activation here. And you get flaccid paralysis because of the inactivation, right? The neurons functioning just fine. What's happening is the acetylcholine can't activate the target tissue because its receptors are being blocked by this particular toxin. I'm going to do two more, I think. Let's do Dendrotoxin. Dendrotoxin is produced by some venomous snakes, probably most famously the mamba snakes. And this blocks voltage-gated potassium channels. And so now we need to think, okay, where are voltage-gated potassium channels? Well, voltage-gated channels in general are from the axon initial segment onward, right? And so, and what is voltage-gated potassium? What is potassium? What's its involvement in the action potential generation? Well, if you recall, the equilibrium potential for potassium is very negative, minus 95 or so. And so if we give it a permeability, it leaves the cell, making it more negative, right? And so potassium is involved in repolarization during the action potential cycle, right? And so while, if I draw an action potential here, while the tetrodotoxin was sort of blocking that portion of the action potential, the up limbs portion of the action potential, because it blocked voltage-gated sodium channels, the dendrotoxin blocks the repolarization wave because that's potassium mediated. And so what happens is the axon gets stimulatory input. The axon can depolarize, but it can't repolarize. And so it stays depolarized. And so if you get no repolarization, this guy stays overstimulated, right? Continues to be depolarized for prolonged periods, causing more calcium influx here, causing more neurotransmitter release, causing more stimulation, overstimulation of our target tissue. So again, back to a rigid paralysis with dendrotoxin. One more. I'm going to squeeze it up in the top here. Botulinum toxin. That may sound kind of familiar, and it does because you've probably heard of Botox, or the abbreviated version, botulinum toxin, right? And what Botox does, this particular toxin, is it blocks the vesicle fusion in the synaptic bulb, synaptic terminal. Right? So what happens is this toxin enters the synaptic bulbs of neurons and basically degrades one of the proteins involved in the vesicle fusion process. Right? For a neurotransmitter to be released, the vesicles have to fuse with this red line, right? The synaptic membrane there, right? The vesicles have to fuse and then exocytose their contents into the synaptic cleft. And what Botox does is inhibits the ability of these vesicles to fuse, so it reduces the release of acetylcholine, right? And so it seems a little bit like the Curare, because while that one blocked acetylcholine binding, this one's blocking acetylcholine release. So same effect, but different target, right? And so evolution has sort of come up with ways to sort of inhibit the, neuronal function of other animals in a multitude of ways, right? So we can really kind of like screw up this neuron's function. And again, linking it to skeletal muscle function or respiratory muscle function is a really good example of sort of how it would be paralytic for animals like a snake, right? A snake bites, a venomous snake bites its prey, and then sort of like backs up for a minute, lets that prey become paralyzed, and then goes in and eats it, right? So that it doesn't risk injury, sort of fighting this animal, that's sort of kicking and biting and everything else, right? So they paralyze it through some form of neurotoxin, and again, we can, inhibit this neuronal function at a multitude of locations here. Last thing to remember is always, whenever you think about a neurotoxin and its effects, don't think about just its effects. Like, for instance, let's do Tetrodotoxin. Tetrodotoxin blocked the voltage-gated sodium channels, so we don't get action potentials. Well, what that means for this cell, the target cell, is if this neurotransmitter opened a ligand-gated sodium channel, it means that we no longer get EPSPs, or we get smaller ones if it's not completely blocked. But we've inhibited the EPSPs on the target cell by inhibiting the ability to generate action potentials in the presynaptic cell. Always think downstream, right? We get the direct action of the direct action of the neurotoxin, but probably more importantly is understanding what the action is or inaction is at the target cell. Do you get a smaller IPSP or a bigger EPSP or whatever it is? And again, that's going to depend upon what type of channel we're binding to at this postsynaptic cell. Again, if it's a ligand-gated potassium channel, right, and you inhibit it, you're inhibiting an inhibitory response, which means you're stimulating. Right? Inhibiting your brakes, something that's inhibitory in your car, would probably make you go faster, not slower. right? And so you have to keep that in mind, is that whole like, there's the direct effect, and then there's the target tissue effect, which will depend upon what type of channels and what type of action we would normally be getting here, whether it's an EPSP or an IPSP, or maybe it's a metabotropic effect, right? We can have all sorts of different varieties of actions depending upon where in the physiology, where in the nervous system these toxins are most active. Okay.